What I'm going to talk about today is part of a larger project that I'm working on. I'm not a nematologist, so I'm working closely with John and Kirsty and people. I come from more of a modelling background and a crop sequencing background, and we're trying to join our work together to see if we can help understand some of these problems a little better. So what I'm actually going to talk to you about is just how the nematodes are affecting wheat, which is basically the summation of some of the work we've done over the last 3 years. For those of you who don't know, we're talking about Pratylenchus thornei. Whenever I say nematodes, I'm talking about Pratylenchus thornei. John and Kirsty will talk about other nematodes, but so a little microscopic nematode reproduces in the roots of susceptible plants. So basically in wheat plants is what I'm going to talk about, and the sorts of problems you see is like, it's not so clear here, but you can see this plot here is looking sort of water stressed. They generally have reduced tillers and a yellowing around the base, and just compared to other crops look quite sick. So the Strzelecki there on the left is a very intolerant wheat variety, suffering quite heavily from nematodes there. The QT8447 is a pre-breeding line that John's team has been working on. It's tolerant and has some resistance. Kennedy is pretty well intolerant, and you can't quite see it, but over here is Wiley, which is probably another of the most tolerant wheat varieties. Okay, so what could be causing this? There's a few things that could be causing it. It could be toxicity to nematodes, it could be competition for crop resources. Those two should probably be crossed out because realistically from a lot of the literature and our knowledge of nematodes in the region, the problems isn't going to occur there. It's probably, most likely thing is it's either the roots being damaged by the nematodes affecting their root length, so the amount of exploration that can go on in the soil, or the function of the root being damaged so that the plants can't, although you have lots of roots, you can't transport water into the plant as efficiently. So what we've done is spent 3 years looking at this. I've had a couple of sites, 2 sites, one which is John's site up near Toowoomba, the Fulmartin site where we've had 3 years worth of trials. The other site we had in 2012 was at Yallarol, and that was run by Richard Daniels at NGA. The difficulty with doing any of this work is it takes a number of years to run each experiment because you have to build the nematode population up before you can run your trial. So it's generally a 2-year, at the minimum, it's a 2-year process. So let's have a look at some of the yields from the first lot of data. And I'll do a bit of explanation here. The varieties are along the bottom and they're in the same order as I told you before. On the left we have the most tolerant variety. On the right here we have the very intolerant variety. The little letters on the top just mean, are the bars different from each other? And it's a little bit confusing there because we have 2 treatments in the sense that the dark green ones are when we planted wheat on a low nematode site. The light green is when we planted on a large number of nematodes. So just to take some of that confusion away, we'll just deal with them one at a time. So dealing with the low nematode site, you can see that it doesn't matter what wheat variety we planted, if there's very few nematodes, there really wasn't a great deal of difference in yield. When we had a high population of nematodes and we planted the trial, we start getting this decline in the intolerant varieties, start losing more and more yield. If we go to Yallarol, exactly the same effect is occurring. You can see we didn't have Gregory in this trial, that's why it's missing. You can see it's starting to show in the low nematodes because the populations build up in the intolerant varieties. So they were getting to the point where you can start seeing they were causing some decline in yield. But basically, low nematodes at the start, very little difference, significant difference in the high populations. Okay, if we start looking at some of the other components of the plant, so leaf area is a measure of the canopy of the plant, the size of the leaves, and it follows exactly the same as the yield. You could basically the same pattern, just different y-axis, which makes sense because biomass determines yield. But if we look at how much water was left in the soil at the end of the season, we can start seeing that the whole thing's flipped around. So those intolerant varieties have left more water in the soil. And if we do a quick calculation on that, so the nematodes have reduced yield, they've reduced the leaf area, they've left more water in the soil. There was about 30 mils of water left in the soil between the intolerant ones and the tolerant ones in the water usage. Now that 30 mils, if we use 10 kilograms as a rough guide, it doesn't matter. You could use 20 kilograms, it still doesn't matter. If we use those, there was 1,200 kilos difference in yield. We can only account for 300, or if you want to double it, 600 kilos, just on water alone. So it's not the fact that they can't get all the water. What I'm saying is it's a fact that the timing of when they get the water. So this is the leaf area over time as the plants have grown. We'll just talk about 2011 first because that's a little bit easier to see. You can see the blue line is the intolerant variety, the black line is the tolerant variety. One's Strzelecki, the blue one's Strzelecki, the black one is the QT line. And you can see that around about 58 to 60 days, the lines start separating. So the canopy, there's more leaf, more tillers, all occurring at that stage. At this point, I'm hypothesising that this is when the nematodes are affecting uptake of water in the plant. The plant is compensating by not putting on tillers, reducing leaf area because it can't get enough water. Even though there's lots of water in the soil, it's going into a sort of drought stress response early on, not producing the canopy. Therefore, it starts producing fewer roots and you get this sort of feed forward mechanism. So it's setting itself up for a lower yield potential. Even though in 2011 it rained every other week, that same effect occurred. So you can't dispel this problem by having more rain or irrigation. 2012 was a drier year. The difference is still there, it's just not as great. If we go underground and just say what was happening to the rate with which the water was being taken up, again the same colour scheme. The black lines are the tolerant varieties, the blue line is the intolerant variety. So this is each soil layer, and this is the rate with which water can be sucked out of those layers by the plant. And you can see that in 2011, the tolerant varieties were taking water out faster. That's because the line is going steeper versus the other lines, which is flatter. So it's taking longer for the water to be taken up. This is partly probably because the nematodes are blocking the roots and causing damage, and also because we've now got less leaf area, so the plants aren't needing to extract as much water. So that's that feed forward mechanism I was talking about. If we look at the rate, so just if you imagine a soil that's filled with water, the speed with which the water is being lowered by the plants taking water up, the tolerant varieties, so the new QT lines or Wiley, are taking water up at around 15 millimetres a day, quite quickly. In 2011, where in 2000 the intolerant varieties can only take water up at about 9 millimetres a day. The point of all of that is that you still have water left in the soil at the end of the season that's not being used and not being turned into grain. Some of that's carrying through to roots, and we can show here that in the lower levels there were roots in the low population of nematodes. There were more roots at depth, again partly because of that smaller canopy, fewer roots, and also because of the nematodes impeding the growth. So, how do nematodes reduce wheat yields? They affect the ability to extract water. They reduce the canopy. The They reduce the root length, though we've got a few question marks there because we can only observe, we can't actually go and measure some of that detail. And we believe it's because the root function's being changed, but we can't go and test the root function, we can only observe. So we know we've hypothesised the processes from what we've observed in the results. So where to now? Or what does it mean for crops? Early damage is more devastating. So planting into a high nematode population is more devastating than planting into a low nematode population. But if you have nematodes, just treat it as if you've got a high population and plant tolerant varieties straight away. Don't try and push it with an intolerant. As these new breeding lines start coming through into the commercial lines, look for the resistant lines because they're what will help reduce or stabilise the nematode population. That's the first stage. Now we're going underground to start seeing what's happening to the population during different seasons. And that's what hopefully we can report about here next year. Thank you. Those of us who have worked with canopy management, the idea that the nematodes reducing early growth, reducing that canopy like we do when we starve with nitrogen to try and reduce early vigour. The beauty with nitrogen is that when you apply it then later in the season or at the ideal growth stage, you can get the growth to come back and you're not getting a lot of big biomass and then running out of water at the end. I guess you do see it a little bit with nematodes, but in the north, because we get the high temperatures coming in that bring the wheat on, our seasons aren't long enough for that to occur. So in the southern areas, there are some stories about the intolerant varieties looking better and doing, equally good yield or even as good a yield under high nematode populations. But it's a really strange mixture of environment.